Okay, so deep inside your bones, right now, there's a silent microscopic boot camp running 24/7. It's training the absolute elite soldiers of your immune system. We're talking about B cells and T cells. And in this explainer, we're going to pull back the curtain and see exactly how your body forges them into perfect defenders.
But think about this for a second. It's a massive paradox, right? Your body needs an army diverse enough to fight any germ you could possibly imagine, but it has to be absolutely certain that not a single one of those soldiers will ever turn on you. So how in the world does it pull off that incredible balancing act? Well, let's dive in.
All right, let's meet the stars of our show, the B cells and the T cells. These are the two major branches of your immune military. And just like any good soldier, they don't just pop into existence. They all start out as fresh recruits, born from these incredible master cells called hemopoietic stem cells, which are nestled deep inside your bone marrow. Now here's the first big split. Even though they're both born in the same place, they go to completely different training grounds to mature and get battle ready. B cells, they do all their training right there at home base, in the bone marrow. But the T cells, they pack their bags and head off to a very special, exclusive academy called the thymus.
Seriously, if you remember just one thing from all this, make it this little mnemonic. B is for bone marrow. T is for thymus. It's that simple, and it's the foundation for everything we're about to cover.
So let's kick things off with the B cells. You can think of their maturation process in the bone marrow as a kind of straightforward but totally essential basic training program. Every single recruit has to go through it, and they have to pass, no exceptions.
And this training happens in 4 key stages. First, as a pro B cell, it literally starts shuffling its DNA around to build one half of its unique weapon, which is its antibody receptor. If it pulls that off, it becomes a pre-B cell and slaps a little test version on its surface. Then it graduates to an immature B cell and gets its first real, fully functional antibody, called IgM. This is where it gets ready for a huge safety check. And finally, if it passes that test, it adds a second receptor, IgD, and officially graduates as a mature, combat-ready B cell. But hold on. Before that immature B cell can be released into the wild, it has to pass that one critical safety check I mentioned. This is a process called central tolerance, and it has one job and one job only: to make sure these newly armed soldiers are not a danger to the body itself. This is the final exam. The immature B cell is paraded in front of the body's own proteins, its self-antigens. If it recognizes and attacks them, it fails, period. It's either ordered to self-destruct, or, if it's really lucky, it gets one last shot to re-edit its weapon. But there are absolutely no second chances for a soldier that attacks its own side.
Okay, so that was the B cells. Now for the T cells. And listen, if B cell development was basic training, then the thymus, well, the thymus is an elite military academy like West Point or Sandhurst. The selection process here is way more intense, way more rigorous, and for a very good reason.
And this analogy is just perfect. Think about it. In primary school, you're mostly just tested on not doing bad things, right? But in a military academy, oh no, you have to prove 2 things: that you are useful and that you are not dangerous. That is the entire philosophy behind T-cell training.
So the journey starts when these T-cell recruits leave the bone marrow and travel to the thymus. When they arrive, they're called double negative because they don't have these key communication tools on their surface, these markers called CD4 and CD8. Then they mature a bit and become double positive, meaning they express both markers at the same time. Now they're ready to face their 2 massive entrance exams.
The very first test is called positive selection. And the question it's asking is pretty simple. Can this T cell even recognize its own side? It has to prove it can read the special ID badges, the uniforms, that all of our healthy cells wear. These badges are called MHC molecules, and they're how our cells present information to the immune system.
And the outcome is brutal. If the answer is no, if the T cell can't recognize our own MHC, it's useless. It can't communicate, it can't get information, so it's just eliminated through death by neglect. But if it can weakly recognize the body's MHC, it passes test number 1. It has just proven that it's useful.
But being useful is not enough, not even close. Now comes the second and arguably even more important test: negative selection. Okay, so we know it can see the uniform, but is it going to attack the person wearing it? Does it bind too strongly to our own body's proteins being presented on that uniform? This is the ultimate loyalty test. If that T cell binds too tightly to a self-antigen, it is dangerously self-reactive, it's a potential traitor, and it is immediately ordered to self-destruct via apoptosis. Only the cells that get it just right, that bind weakly enough to show they can see the uniform without attacking the person, only those are allowed to graduate. After surviving this incredibly tough two-part exam, the T cell finally graduates and gets to pick a specialty. It gets rid of one of its markers, depending on which type of MHC ID badge it got good at recognizing. If it recognized the Class II badge, it becomes a CD4+ helper T cell, kind of like the field general that coordinates the entire attack. If it recognized the Class I badge, it becomes a CD8+ cytotoxic T cell. So let's just quickly recap the big differences here. B cells train in the bone marrow, and they only really have to pass one test. Don't attack yourself. T cells, on the other hand, go to the elite thymus academy and have to pass two tests: prove you're useful, and then prove you're not self-reactive. This whole two-step process is why T cells are called MHC restricted. They are literally trained to only listen to our own body's cells.
So, why does all of this complex training matter so much? I mean, why go to all this trouble? Well, because when this system fails, the consequences can be absolutely catastrophic. This isn't just some abstract biology lecture. This has profound, real-world effects on people's lives.
Take, for example, DeGeorge syndrome. This is where a person is born without a thymus. No military academy means no trained T cells, and that leads to severe recurrent infections. Or think about autoimmune diseases like lupus or rheumatoid arthritis. That's what can happen when negative selection fails and those traitor cells escape into the body. And in the most severe cases like SCID, often called bubble boy disease, the genetic machinery that builds the weapons in the first place is broken. No receptors can be made, so no functional B or T cells can ever graduate.
And so you see, it all comes down to two sides of the same coin, two goals for the same incredible system. On one side, you have to generate almost infinite diversity to create an army that can fight any enemy, known or unknown. And on the other, you have to enforce absolute, unflinching self-tolerance to make sure that incredibly powerful army never, ever turns on itself.
So, the next time you're just going about your day feeling perfectly healthy, take a second to remember why. It is not an accident. It's because deep inside you, this silent, relentless, and unbelievably precise training program is running around the clock. It's forging an army of perfect guardians, where every single one has passed the ultimate test, to know the difference between an enemy and you. It is the most important military operation in the world, and it's happening inside you right now.